Welcome to this talk today in the lecture series that we called Halmstad Colloquium, which is a series of distinguished speaker talks by international guests. It is sponsored by the School of Information Technology and its research environment called Halmstad Embedded and Intelligent Systems Research. And today's talk is given by Professor Jorge Armony from McGill University in Toronto. Sorry, we talked about Toronto during the lunch in Montreal, Canada. And Jorge is a world-leading expert on neurobiology of emotions and emotional processing. He's a professor at McGill University, connected to several departments, as I understand it, both psychiatry and psychology, and also the Department of Neurology and Neurosurgery. And he's also active as a researcher at the Douglas Mental Health University Institute and at what's called the International Laboratory for Brain, Music and Sound Research, BRAMS. I hope it's correct, Sean. Yes. Yeah, it says, okay, there it is. Jorge Armony has studied and done research and taught at several universities all over the world. Actually, his first degree was in Buenos Aires, that was then in engineering, or physics, sorry, in physics. And then at New York University, he took his PhD in neuroscience. He has been a postdoctoral fellow at Oxford University and at London, University College London. And he has been a guest researcher, a guest professor at universities in Paris and in Mexico City. And now he's, as I mentioned, professor at McGill University in Canada. I have had the opportunity to look through his CV, and I found that you have received many research grants. I counted to more than 20 over the last 15 years or so. So you've been very successful there. And George has also authored or co-authored between 40 and 50, if I counted correctly, journal papers. And looking at the citations of those, there seem to be well over 10,000. So it's really research that has become read and cited all over the world. So we are very proud to have you here today, George, and looking forward to your talk. Welcome.
Thank you very much for the introduction and for the invitation. It's my third time in Sweden. Last year I was also an opponent in Chalmers. In fact, my first time in Sweden was 25 years ago by mistake. I can tell you later. I was planning on going to Copenhagen, and for some reason I took the wrong boat. I ended up in Sweden from Poland. But so now I'm here on purpose, and it's nice. So what I'm going to do, it's a strange talk in a way, because my understanding is some of you, or many of you, or most of you, or all of you are more from the computer science side of things. So I wanted to kind of guide you through some of the ideas we had over the years of how we started doing some computational modeling and how that led us to do some research in humans and taking it all the way to psychiatric patients. I'm going to do that, as you imagine, reasonably quickly. I'm going to go over time as usual, so you can stop me whenever you think I've said enough. And one of the things, whenever I give a talk, or whenever any of us doing research on emotion give a talk on emotion, particularly for audiences that are not that familiar with emotion research, the assumption, so we have kind of to start saying all the things we're not going to talk about and the things that we don't do research on. Because when you think about emotions, you think about a lot of things, and typically people associate emotions with feelings. And so you're going to study feelings, and that's very complicated, and how you do that, and so on and so forth. And that's clearly an important area of research, but that's not the area of research that I'm interested in. I'm studying. I'm studying, you can think about it as information processing of certain types of information that has a particular meaning, a particular value, which is this survival-related or affective biological salient value, right? And that's, but we can apply all the techniques and all the approaches that we do with other things like vision or audition to answer these questions. And so one of the most studied emotions for many reasons has been fear. And I'm going to start a little bit with fear. I'm going to move on to other sort of nicer emotions. And the idea behind studying fear is, first, obviously, it's a easy emotion to induce. It's a very ubiquitous emotion. And even since Darwin, this has been recognized that emotions in general, and fear in particular, is essentially the same across species. And in fact, Darwin used fear and emotion as one of the fundamental sort of arguments for his theory of evolution. And so this is a citation from his book, The Expression of Emotion in Man and Animals, and where he says, with all or almost all animals, even with birds, terror causes the body to tremble, the skin becomes pale, sweat breaks out, and the hair bristles, as is known to be the case with man, and as I've seen with cattle, dogs, cats, and monkeys. So he's already saying that the reaction, these fear reactions to a threat, are universal in the sense that we all experience the same physiological responses. Some of the responses are more species-specific, but a lot of them, particularly more at the more physiological level, they seem to be shared by all or most species. And in fact, Darwin didn't know this, but even the neural substrates of emotional processing, in particular fear, seems to be fairly similar across species. So the key player here will be the structure known as the amygdala. We'll see a little bit more about it in a second. But the amygdala, which is this little structure located in the temporal lobes, is essentially the same for rats, monkeys, and humans. It has the same structure in terms of both the cytoarchitectonic, that is the structures of the neurons, the connectivity, but also the function. And in a way, the idea is that it's a system that is very old evolutionarily, that's worked very well, so there's no need to change it. So even you can go back to birds and fish, and they don't have amygdala, but they have structures that are similar to the amygdala in terms, again, their function. The structure, as I always say, the smaller the animal, the longer the name, but the idea is the same. In fact, this is a section, and what's in black is a study where they lesion this part of this long-term structure that is similar to amygdala. And if you do that, the little goldfish have no fear anymore. They don't undergo fear conditioning. So it's not only structurally that is the same, but functionally it seems to be the same. And so this makes it the perfect sort of model for studying cross-species processing emotions. So what you study in experimental animals, particularly in rodents, you can very easily translate to humans, and some of the hypotheses that you can sort of have with humans, you can test them in other animals and so on and so forth. And that's why it's been a very sort of rich area of research across different domains and techniques. The amygdala is an interesting structure because it's very small. It has its volume in humans about 1 to 2 cubic centimeters, so it's something like this small. And it has 12 million neurons. That's essentially nothing for the brain. And the main input to the amygdala, which is the lateral nucleus, have about 4 million neurons. And the main output of the amygdala, the central nucleus, which is the one that sort of governs all your physiological and behavioral responses, has 350,000 neurons. That's essentially nothing. So it's a very small structure. It still is very complex. It has about a dozen of nuclei, and some of those nuclei have subnuclei, and those subnuclei have sub-subnuclei. In fact, some of our early work was looking at the ventral aspect of the dorsal aspect of the lateral nucleus of the amygdala and showing that the neurons there behave very differently from the neighboring one in the ventral aspect of the dorsal aspect of the lateral nucleus of the amygdala. So even with the very, very small structures, with very few neurons, they're very different in their behavior. And that's what makes potentially the amygdala such an interesting structure. The other thing that makes the amygdala important in a way is location. location, location, location. The amygdala is connected to everything. In fact, there's been a map done by recently, first of the visual system, but then they did it of the whole brain. And the structure that is most connected to almost every single other structure in the brain is the amygdala. The amygdala is at the center. It receives information from every sensory modality. It receives information from regions like the hippocampus that provides memory and context. It receives information from the prefrontal cortex that's more in terms of inhibition and planning and sort of more executive functions. And then it has outputs downstream to again, physiological and behavioral outputs that allows you to respond, your heart to start sort of racing, your muscles to contract, your blood pressure go up, et cetera, but also has feedback connections to all areas of the brain, cortical and subcortical. In fact, it projects back to regions where it does not receive information, like the primary visual cortex. So the primary sensory areas don't project directly to the amygdala, but the amygdala projects back to them. So it's in a position to modulate behavior and modulate perception and modulate attention and modulate memory, decision making, and so on and so forth. So that's one of the things that makes it fairly unique as a structure. So this is a model that essentially a number of researchers, particularly Joe Ledoux, who's my PhD supervisor, and others, built based on mostly the auditory fear conditioning in the rats, but it's been shown to apply to a large extent to humans and other modalities. And so given emotional stimulus, as a stimulus has some emotional value, first we'll go through the sensory processing. So you'll go to the sensory thalamus. In this case, is the medial geniculate body because it's the auditory thalamus. In the vision would be the lateral geniculate nucleus. And from there, your traditional information path would be that it would project to primary sensory cortex and to higher unimodal cortices and so on to other higher order cortices and hippocampus and prefrontal cortex and so on and so forth. And that's true. And as I was saying, the primary sensory cortex does not project direct to the amygdala, but all the other sensory steps do project. So the amygdala receives a cascade of information of different levels of processing, right? And they all converge into the lateral nucleus. What makes also interesting has been sort of a quite active area of research, and that was first described by Joe Ledoux, is that the thalamus also projects directly to the amygdala. So there's a direct monosynaptic connection between this part of the so-called extralemniscal area of the thalamus, which is a less tuned part of the sensory thalamus, and it projects directly to the lateral nucleus of the amygdala. And not only so you have in the same nucleus projections from different areas. In fact, some of the same neurons receive projections from the thalamus directly and from the cortex. So these neurons are in a position to integrate information of different levels of complexity and at different times, because clearly you can imagine a monosynaptic connection is much faster than a polysynaptic connection. There is a connection that goes through different sensory cortical steps. So you have these different pathways, and this has been sort of known as this is the direct subcortical pathway and the cortical pathway, and Joe calls this kind of the quick and dirty road, and this is the slow and highly processed, detailed road. So you have this type of scenario where you have information reaching the amygdala both in a direct and fast fashion, and it's sort of more detailed but slower fashion. So this, back in the day, done more than 20 years ago, in collaboration with David Servan-Schreiber and Jonathan Cohen, when both were at the time at Carnegie Mellon University in Pittsburgh, we developed a computational sort of connectionist model, embarrassingly simplistic, but it was arguably the first, or one of the first models that were trying to not only explain behavior, but also be based really on the anatomy of the system and the physiology of the system. So we tried to replicate as much as we could with the little knowledge we had of the fear conditioning pathway, and try to see if we could mimic some of the behavior, not only at the output, but also within the system. And I'm not going to go into details, but the model was reasonably successful at showing the behavior of neurons, neurons typically in the auditory domain, but also in the visual, have what's known as receptive fields. So they respond to different frequencies, auditory frequencies, and they have a best frequency. They prefer a given frequency. And some are more finely tuned than others. So this part of the thalamus has a very, very tight tuning curve, so it responds mostly to one frequency and its neighboring frequencies and nothing else, whereas areas of the so-called extralimniscal thalamus have a more broad receptive fields, that is, they respond to more frequencies. And one of the interesting things that has been observed experimentally, and we were able to sort of reproduce this with a model, is that when you do fear conditioning to a particular frequency that is not the best frequency of the neuron, there's actually a shift in the receptive field of the neuron, and that frequency becomes now the best frequency of the neuron. So neurons actively change their tuning properties to tune to the new stimulus, which is, or not new, but to the newly important stimulus, which is the conditioned stimulus. And we show that some of them have this behavior and some of them don't, and this is similar to what's been shown in the electrophysiological experiment. And we also sort of showed that the behavior was very similar, both in the learning rate and also in the fact that if you conditioned, so you do classical conditioning, I don't need to explain to you what classical conditioning is. You take a stimulus that is meaningless, a sound, a color, a picture that has no meaning, and you pair it with an unpleasant aversive stimulus, can be an electric shock, can be a loud noise. And then after a few pairings, sometimes it only takes one, this condition, the so-called conditioned stimulus, become in itself aversive. And you have these conditioned responses, these defense responses. To that, you can do it in the appetitive side, so you can do it to reward, but that would be reward conditioning. And it was first described by Pavlov and his dogs and so on and so forth. And so when you condition to a stimulus, you have these responses to this conditioned stimulus, but also you have some level of generalization. And in the frequency domain is what's known as stimulus generalization gradient. That is, you will show responses, fear responses to the conditioned stimulus, but also to its neighboring frequencies. And that makes sense evolutionary speaking, because if you were to encounter something, a sound that is associated with a particular bad event, and you hear the same sound but it's slightly off, but it's not exactly the same frequency, it's not very safe to say, well, no, it's slightly out of tune, so I shouldn't be afraid, right? So, you're better off generalizing a little bit, not too much. So, in fact, we tried, with this idea, we tried to see what would happen. What were at the time, one of the questions, what was the difference between this so-called slow and detailed pathway and this fast and quick and dirty pathway? And one of the ideas was that the slow pathway, because it goes through the cortical steps, has more processing capacity than the subcortical pathway, has very limited capacity. So what we assumed is that if we were to lesion the cortex, either in real individuals or in the model, what we would get is a sort of catastrophic generalization, we call it. That is, that the tuning, because the tuning curve of the projection of the areas of the thalamus that project to the amygdala and those of the amygdala themselves are very broad, we expected that the stimulus generalization would broaden if the cortex was not present. That is, if the system could only rely on this quick and dirty pathway, there should be a wider generalization to frequencies, to stimuli that are similar to the original conditioned stimulus, but not the same. So we ran first through the model, and that's not what happened. We found that in the simulations, lesion in the cortex had no effect on the generalization gradient. So we thought that we had done something wrong with the model, and so we decided to test this on animals to see what we had done wrong. And it turns out that the animals behave exactly the same. That is, there was no effect of the lesions of the auditory cortex in generalization gradient. And this was, we could show in the model that this was due to some sort of population coding. That is, although each individual unit had very broad tuning, all of them put together could have much better tuning than each of them separately. So this has been shown in the motor system as population code. Right? So this was nice in a way, sort of to see that the model was actually able to predict behavior in a way that we hadn't anticipated. It allowed us to explain that behavior because we could look inside the model easier than we could look inside the animals. But sort of brought us back to the question of what this subcortical pathway was doing, if anything. At the time was a sort of an active debate, people saying that the subcortical pathway was just the appendix of the brain. That is, was something that at some point was useful, but it was no longer useful. It was, you know, we were making just a big fuss of something that wasn't really doing anything. So we were trying to understand a little bit what could be the difference. And one of the things that didn't seem to work was cortex is very good at processing stimuli and isolating stimuli that are similar. So this is what we thought it didn't work. The other thing that the cortex is very good at doing is modulating attention, telling you what's important in the world and what's not. So we live in a crowded environment, and we're very good at filtering out all the relevant information and focusing of what we're interesting. So you're all paying attention, fascinating by my talk, and you can ignore sort of things that happen around. If somebody comes late and they come through the back and they will sit because they're not important, they're irrelevant, then you can filter out that information. In fact, your cortical system is actively reducing the representation of those stimuli. So we thought there might be something there, and in fact, at the time, it was brought, David Servan-Schreiber, remember something from his thesis supervisor that was Herb Simon, a mathematician, psychologist, Nobel Prize in economics. And Herb Simon had written a paper in 1967, basically saying that if you want to build an intelligent system, it has to have emotion. He said there are two requirements for any sort of organism to be capable of survive. One is to have a goal-terminating mechanism permitting goals to be processed serially without one monopolizing the process. That's what cognitive science and particularly computational modeling have been focusing on, goal-directed behavior. Sort of system that have a goal and could reach it, and could have competing goals, and as he's saying, there has to be an ability to serially go from one to the other. But he said a system that with this would not work. A system, in order to work, needs a second system, which is an interruption mechanism that would have the properties, I said, usually ascribed to emotion, allowing the processor to respond urgent to urgent needs in real time. So he proposed that something they call the interrupt system. And the interrupt system was something that had to go on continuously and work substantially in parallel. So you had to pay attention to the environment regardless of what the organism is doing, regardless of what the goal at that time is. There has to be a system that is monitoring the environment in parallel, and it has to be capable of interrupting whatever is going on, setting aside ongoing programs to attend to that urgency. So the idea is you might be thirsty or hungry, so you're looking for food, and that's your goal, and it's a survival related goal. But if a predator is about to eat you, if you're about to become somebody else's lunch, you should hold off to getting your own lunch and get out of the way, right? Because otherwise, there's no point in getting lunch. So there has to be, so you're focusing, your goal is very clear, you're looking for food, but you have to have a system. So your cortical system will filter out the rest of the world. Well, that's very good until something dangerous comes along. So you have a system that works in parallel and is able to detect this potentially dangerous stimuli and interrupt the ongoing system and redirect the resources to that. So we thought that maybe that was the whole point of this subcortical pathway. The subcortical pathway would be less sensitive to attentional modulation, and it would be able to work in parallel of the other system. And even if it's a rough system, it's okay, because what it needs is to detect potential threats and redirect resources to that source, to that part of the world that has the potential threat, and then let the cortex do its job of telling you. So the idea, the typical example that Joe would give is you're walking in the woods and you see something wavy on the ground, and you respond because it may be a snake, and if it turns out to be just a branch, then it's okay. But it's better to respond to that branch than not at all. By the way, Ledoux is snake phobic, so he particularly likes that, uses that story. So we decided to modify our model, make it a little bit bigger, and incorporate some more sophisticated or slightly more sophisticated cortical systems to include an attention component to try to test that. We kind of did it not very successfully. And we wanted to start testing this. And at the time I was finishing my PhD, and also I had come to the conclusion that have rats pay attention to things is very difficult. I mean, they pay attention to things, but it's not what you want them to, and you have no idea what they're doing. So decided to sort of go up the evolutionary scale and try to deal with animals that are slightly more easy to control, and at least you can sometimes talk to them. So decided to move to humans. And so this is just to give you a sense of where the I mean, as I said, it's the same, but where the human amygdala is. It's a very small structure located just in front of the hippocampus. You may be more familiar with the hippocampus as the memory area. And perhaps no chance that the amygdala is so close to the hippocampus, and they're heavily interconnected, and emotion and memory are very important to go together. So, in fact, the amygdala in the humans was described by Carl Burdach in the 1800s. This is a real brain with the bottom part sort of chopped off, so you can see the amygdala. It's eye-colored. This doesn't come like that, and the real brain. Amygdala stands for almond, by the way, although I always say that it's more like looks like a Cajun nut than amygdala. And the other twist and for the studies in humans is that rather than using fear conditioning, although we did some studies, we decided to switch a bit the stimuli and to use facial expressions. Facial expressions are very important for social interactions. It's very useful to know they can tell you a lot about the people you're interacting, so I can sort of tell, whether you're falling asleep, you're really excited, you're really bored, or you're, all looking at Facebook in your computers. And it allows me to adjust my behavior and even predict predict your behavior, and therefore adjust my behavior based on your predicted behavior, right? But also they can signal important information about the environment. So a fearful face, and this is always a question we get asked, well, a fearful face is not the fearful stimulus, it's not a threatening stimulus, not in itself, but it's telling me there's a threat somewhere. There are two options. If I see somebody with a particularly conspecific, somebody from my own species, afraid, there are two options. Either this individual is afraid of me, which is a good thing, or he's afraid of something else, which I should probably be afraid of as well, because we share the same predators. I say, well, we share the same dangers. So knowing that somebody is afraid is very useful. And so emotional expressions, as Darwin was saying, are shared across species, and even potentially one could argue that facial expressions are also very easy to recognize in other animals. Right? So I sort of told you already why it's useful or important to study emotional expressions. So social interactions, they arguably are universal. That was work by Paul Ekman, which, by the way, that's Paul Ekman, showing his famous emotional expressions. This has been debated, so in recent years. They may be innate, again, that's debatable, but at least we know they're acquired very early in life. If they're not completely innate, hardwired, at least babies are able to recognize basic emotions, particularly from their mothers, and particularly the stereotypical emotions, sadness and happiness, they can pick up very, very easily. And perhaps also it's important, this has a clinical application, because almost every single psychiatric disorder will be associated with some deficits in the expression or perception of emotional expression. Some are particularly, this is a particular important symptom, in others less, but if you look at the literature, every single psychiatric disorder you can think of, there'll be a study showing that they have deficits in emotional perception or expression. And then the other slightly different thing is, in order to study brain processes in humans, it's more difficult to use the type of techniques that you would use in experimental animals, namely lesions and depth electrodes. So we use other techniques, the so-called neuroimaging techniques, and particularly the one that we've been using for the past few years, or a couple of decades now, is magnetic resonance imaging, functional magnetic resonance imaging. So I'm gonna tell you much about that because we don't have time. We can talk about it later. But essentially, just to give you, so the brain is essentially water, right? And water is essentially hydrogen. And hydrogen is it's an atom that has because there's only one proton in its nucleus. It has a spin, has this quantum mechanic feature known as spin, and spin has magnetic properties, so interacts with the magnetic field. So if you put a bunch of spins in a magnetic field, they will tend to align in that field, and you can perturb those spins. They will go back to their original position, and the speed which they go back to to be aligned with the magnetic field depends, among other things, on where they are. So depending on the magnetic density, in a way, of the medium, they will go faster or slower to their original position. They will recover at different speeds. So if you measure that so-called relaxation time, it will tell you in which medium they're embedded. And so you can create, by putting that thing in a particular machine, namely a magnetic resonance system. This is, by the way, in physics, it's called nuclear magnetic resonance, NMR, and it's just that the word nuclear is not very friendly, so it's been changed to MRI. So you can create magnetic maps of any object, and then you can translate those magnetic maps into actual maps of things, and so you can take essentially pictures of the brain. So you can use different types of measures. So you can have the so-called T1 weighted, so use the so-called transverse relaxation, and you get what's known as structural image, and you can use the density weighting. So these are typical ways of looking at the brain, but these are ways of looking at the structure of the brain, which is very important, but we want to look at the function of the brain. So we want to see something online. And the way to do that is to take advantage of the properties of hemoglobin, the blood. And hemoglobin turns out to come in two flavors. You have oxyhemoglobin, that is when the hemoglobin is bringing the oxygen to the cells. And when it's doing that, oxyhemoglobin happens to be a so-called diamagnetic substance, which is mildly repelled by magnetic fields, but essentially is transparent to magnetic fields. So if you put a drop of blood that is mostly oxygenated blood, that is arterial blood, in a magnetic field, there's no disturbance, there's no interaction between the blood and the magnetic field, so it's essentially transparent to that. On the other hand, when the hemoglobin already deposited the oxygen and is going back, so it's deoxyhemoglobin, it becomes, there's a conformation change. The molecule, in a way, becomes three-dimensional, has a dome feature, so it becomes paramagnetic. That is, it becomes, it acquires a magnetic moment that is now non-negligible. It's a substantial magnetic moment, so it interacts with magnetic fields. So if you put now a drop of blood that is mostly deoxy, that is venous, it will disturb, it will interfere with the magnetic field. So what you can do is you can measure the amount of interference in the magnetic field, and so you can see a relation between oxy and deoxyhemoglobin. And why is that important? Well, the reason, and that's been known since the 1800s by Sherrington, a neurophysiologist in England, that when you have an increase in neuronal activity, because neurons then are firing their action potentials, releasing neurotransmitter, they get hungry. They need to replenish their energy. And the way they replenish their energy is through oxygen, mostly. A little bit of glucose, but mostly oxygen. So when you have increased neuronal activity in that part of the brain, the circulatory system knows that that part of the brain needs more oxygen. So there's an increase in blood flow and blood volume of oxygenated blood. And as I say, so the circulatory system is a little bit like a Jewish mother. If you need 10, it will send 1,000, right? So although some of the it's okay, I'm Jewish, so I can say that. It will send the amount of oxygen, but more. So there'll be an overflow of oxygenated blood, which means that relatively speaking, the deoxy content will go down because there's an increase in oxyhemoglobin. And because the deoxy is the bad boy, because the deoxy sort of is the one that has the paramagnetic properties. So if you decrease deoxy, you decrease the interference, and therefore you increase the signal. So the bottom line is you have more neural activity, you have more signal. And the signal is called BOLD because it stands for blood oxygen level dependent, because it measures blood oxygenation in the brain. So essentially, this signal becomes a proxy of neural activity. So we're doing an indirect measure of neural activity, but it's still a measure of neural activity. Okay? So just, again, I'm spending a bit of time on this because it's important to understand this, to know what we're talking about, although even I'm going to go way over time. So imagine I present, there are a couple of things that are important to keep in mind. One is, so let's say I present a picture of a house and I get this. This is the front of the brain, this is the back of the brain. So you have stronger signal in the back of the brain because of the visual system. That in itself doesn't tell us anything because I need relative levels of oxy to deoxyhemoglobin. So I need to compare. So what I need to do is to have another condition. So let's say now I have present a picture of a face. And if you look at these things are very similar, and that makes sense because they're both visual stimuli. So what I need to, if I want to see the difference between these, I literally will take, I take, this is like an image, like a regular digital image. So I have pixels, we call them voxels because they're three-dimensional pixels. And we just literally take the one minus the difference, one minus the other. And obviously, this doesn't tell me anything. I need to do statistics. So what I do is I simply divide the difference by the standard error of the mean. And I get what's called a t-map. A t-map is simply a bunch of t-values. Each voxel has a t-score. So I have 100,000 t-scores. Most of them are not significant. So instead of publishing a paper with 50 pages of voxel saying NS, 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 and paying thousands of dollars for page charges, what I do is I say, I'm going to get rid of all the voxels that are not significant because they're not interesting. So I'm going to put a threshold, 0.05 or whatever you like now that it's being in the debate. So in this case, I use what's called a family-wise error corrected for multiple comparisons, 0.05. And all it survives is the little drop there. It's very hard to know where that is. So what I do is I also took a T1, a structural image of the same brain, and they're co-registered. They're in the same space. So what I can do is I put one on top of the other. And I show what's known as an activation map or SPM, statistical parametric map. So when I'll show you a few of these things, there are a number of things to take into account. There's no real data in here. This is a statistical map. So there's no physiology, there's nothing. This is not signal, it's statistics. The second is that the fact that there's nothing there doesn't mean that the brain is not responding. It means that there's no there's no statistical difference between conditions. Right? This is a question that happens regularly often. I show this and say, this is the part of the brain where responds in this case to houses. And somebody say, well, that's odd because there's no visual cortex response. How does the information get there? Yes, there is. It's just that it's the same amount statistically between faces and houses, so it cancels out. Okay? That's important to keep in mind. So, for example, if I compare faces to houses, I get this here usually is bigger, but I get this little thing in the ventral temporal lobes, which is the fusiform gyrus, and this is so-called the face fusiform area that described originally by Nancy Kanwisher. It's the face area of the brain. If I do the opposite, I get the parahippocampal area, that is the house area of the brain. The parahippocampus is involved in spatial navigation, so it makes sense. Okay, so you can do a lot of these things, but let's go back to this idea of attention that I was getting to. So, how can we play with these things? So, imagine from what you learned today or what you knew from before, if I were to do a contrast, these things are usually known as contrast, between this and this, I should get 0. This and these are exactly the same stimulus. So if my design is fine, if I compare this to that, my SPM should be blank, should be nothing. And indeed it is. But I can play with this. I can play with the attention focus. I can tell the subject when it's in the scanner to either pay attention to the horizontal stimuli or pay attention to the vertical stimuli. And I can present these faces and houses randomly, either in the horizontal or in the vertical position. And by doing that, I can present this stimulus once when they are paying attention to the houses, and I can present it later in the experiment when they're paying attention to the vertical stimuli, and in this case to the faces. So I'm presenting exactly the same stimulus, but in one case they pay attention to the houses, and in one case they're presenting attention to the faces. Obviously, I can do the reverse. And if I do that, what I get is activations in exactly the same areas. That is, I get significant activation in the fusiform area or in the parahippocampal area to the exact same stimulus, but as a function of their attention. So this is what I was telling you, that not only when you're paying attention to a particular stimulus and ignoring the others, not only you're doing it behaviorally, but your brain is doing it, obviously. And by doing that is enhancing the representation of the attended stimulus and decreasing the representation of the non-attended stimulus. And it's working very well, as you can see here. In fact, if I ask subjects, if I interrupt the study at some point, and I ask subjects, I say, well, you just saw faces and houses. I'm going to ask you questions about the faces. I'm going to ask you, was it a man or a woman? Assuming that the two faces were the same. Right? Was it a fearful or a neutral? And was it, I'll show you two pictures, was it John or was it Jim? Right? If in that trial you were paying attention to the faces, you're 100%. It's a very simple task. If you were paying attention to the houses, you're a chance. So this is in a way what's known as inattentional blindness. You see the stimulus. We're not saying that you're not seeing the stimulus, but you're not actively paying attention, so you're a chance at knowing things about the stimulus. Doesn't mean you haven't processed it, but your awareness of the stimulus is reduced. Okay? So now we have a manipulation that with which we can manipulate, modify attention. We can tell people to pay attention to something. We present the stimulus, I didn't tell you, but for about 200 milliseconds. So they're very fast, so it's very hard to do this. And we do this, and we added a twist. So the faces now can be either neutral or emotional, fearful. So now we have what's known in the trade as a 2x2 factorial design. We have faces can be attended or unattended, and faces can be neutral and fearful. And the question what we're interested in is what's going on with the interaction. Is there a difference in the processing of fearful faces when you're paying attention to them and when you're not paying attention to them? If we focus on the fusiform face area, that is that part of the brain that responds and likes faces, what we see is first, as I was showing you earlier, there's a main attention of attention. There's a main effect of attention. So attended faces have a stronger response than unattended faces. There's also a main effect of emotion. There's an enhancement of the response to fearful faces than to neutral faces in the cortex. But it seems to be sort of an additive effect, right? So the strongest response is to fearful attended faces, and the weakest response is for neutral unattended faces. That makes sense. What happens in the amygdala? The amygdala doesn't care about attention. The amygdala responds to fearful faces equally, regardless of whether they are attended and unattended, suggesting that indeed this idea that Simon had about this interrupt system, we cannot prove here that it is due to the subcortical pathway in any way, but what we're showing here is that indeed the amygdala might be playing at least part of that role of this monitoring parallel mechanism that Simon was proposing, in that whether you're paying attention or not, the amygdala is responding to potential threat. Okay. One of the questions that comes up is, well, but we know that the cortex projects to the amygdala, and the amygdala projects back to the cortex. Although the cortex is modulated by attention, it's responding more to fearful than neutral faces. So how do we know that the amygdala is really doing anything and it's just the output of the system? Is it really doing anything, or it's just receiving the information from the cortex? Well, the way to do that is one way to do that. So if you take healthy controls, if you take normal people, students, so they will, and you compare fearful to neutral phases, they will have a significant activation in the amygdala, and here is a side view, but it's the fusiform phase area. Right? So what happens if you take now individuals with no amygdala, or at least with less amygdala than healthy individuals? In this case, you take individuals who suffer from temporal lobe epilepsy and had amygdala damage due to a temporal lobe epilepsy, which is a common case they have, what's known as amygdala sclerosis. In these individuals, they have no amygdala, so we don't expect an amygdala response to fearful phases. The question is, do we still get a fearful response to response to fearful phases in the cortex? And the answer is no. So it's not that these subjects are what's known as prosopagnosis. It's not that the subjects do not recognize phases. And it's not that the subjects do not recognize fear, but the enhancement of the representation of fearful phases in the cortex is gone. So this strongly suggests, and in fact, the degree of goneness, the degree is related to the degree of amygdala damage. So this strongly suggests that the enhancement of the representation of fearful phases in the cortex is due to the amygdala, due to feedback from the amygdala and not the other way around. Okay? So this is what this study doesn't prove, but it suggests. Right? And so this type of there's been hundreds, if not thousands, of studies using this type of stimulus, and it's been shown to be useful also to study psychiatric population. This is just an example of work by other people and by us, showing enhanced in post-traumatic stress disorder, which is a disorder in which somebody who's been exposed to trauma then responds, has exaggerated response, strong responses to reminders of the trauma, even if they are completely unrelated in times and space compared to the original trauma. And what several studies have shown is that these individuals have an exaggerated amygdala response, not only to the trauma reminders, which would make sense, but to any other stimulus, so particular fearful phases. So if you take, for example, these are Vietnam vets, and these are people who had a car accident. And so fearful phases have nothing to do with their car accident, yet they have an enhanced response to fearful phases, particularly in this case, in both these cases, to masked fearful phases. That is, fearful phases are presented subliminally with very little, no or very little awareness. And this is a correlation with their symptom severity score. So not only do they respond more strongly to the stimuli, but the worse their symptoms, the stronger the response, suggesting that this hyper activity of the amygdala is not just for the trauma reminders, but seems to be a general aspect. Whether, obviously, the million-dollar question is whether it's a consequence of the trauma of PTSD or it's a risk factor. And there's some studies suggesting one is one and others, but this is a type of work that's been done. Okay? By now, you should be really convinced that, fearful phases are the way to go. And that's great, but if an alien species were come to visit our planet, and all the information they had was from Medline, from research papers, they would think that humans are essentially that. Right? They would think that we're basically floating faces with emotions, because that's what the literature is all about. It's about facial expressions. But there's more to that. Right? Even in the visual domain, you have other ways of expressing emotion. So even if in this individual you don't see the face, you sort of know what his state of mind is. Right? If this is your supervisor, your boss, and you're about to walk into his office, and you see that, you may think twice before coming to going to tell him that you forgot to submit your fellowship application or the paper was rejected. Right? So there's more information that you can convey, even in the visual domain, done by faces. So the question is, what happens with those type of stimuli? So the first one is, we know, or I told you that there's a part of the brain that likes faces, the face fusiform area. It's been debated whether it's innate or not, but there's a face area of the brain. Is there a body area of the brain? And the answer is yes. There's a part of the brain now also in the visual system, in the so-called lateral occipital complex, that responds more to bodies to body parts than to other types of stimuli, including faces. And this has been known or named as the extrastriate body area. So we have the machinery to perceive or to process body information. Moreover, work by Julie Grès and Bea de Gelder, and Swan Pichon was a student of them, and now is a researcher in Geneva, shown that the amygdala also responds to body expressions of anger and fear compared to neutral. And in a study that we did, we showed using now videos of anger expressions. You can show that the amygdala responds to body expressions of anger if they're intense anger, whether they are being, I was telling you earlier, whether, when somebody is afraid, can be afraid of you or somebody else. Well, somebody is angry is the same story, but the opposite. Somebody might be angry at you, which is very bad news, or can be angry at somebody else, which is, okay, too bad for the other guy. So we could expect that the amygdala would respond differently to this type of stimuli. Well, if the anger is extreme, it's very strong anger, the amygdala doesn't care. So this is self, so it's anger directed at you, and this is other, anger directed at other. There's no difference. The amygdala responds to both. But if there's less anger, if there are more subtle levels of anger, you see that when the anger is directed at you, there's an increased sort of response associated with increased levels of anger. But if it's directed to others, there's no response to subtle anger, suggesting that the amygdala does differentiate between what we can call self-relevance. If the emotion of the action is relevant to you, then it responds. And one could argue when somebody is really angry, whether he's angry at you or somebody else, is relevant, because once it's done with the other person, you might be next, right? So it makes sense. In fact, in the face literature, we have something similar. In the face, you can have this with eye gaze. So what some people in DI and Patrick Villemier, I forgot to tell you that the attention study was done with Patrick, who's now in Geneva. You get an amygdala response to subtle, if it's strong, fearful or angry expressions, the amygdala responds. But if they're more subtle, the amygdala will respond to anger directed at you with eye gaze directed, or fear directed elsewhere. And that, again, makes sense, because fear with gaze directed elsewhere is telling you that there's a threat somewhere else. And anger directing at you telling you that there's a threat right in front of you. So these two are self-relevant, and the amygdala responds to those. So the amygdala does detect or differentiate between these two conditions. Okay? So things like this engage the amygdala. But there's something missing. There's another way of expressing emotions in another modality, in the auditory modality. And in fact, some people will argue that auditory emotional expressions are more efficient than visual ones. So as I said, if you wanted to help that lady using only visual information in order to know that she's, I'm going to spoil it for you, but she's in danger, you had to be in the shower with her. Whereas in order to detect her fear through the auditory acoustic domain, you could be anywhere else. Right? So in fact, the acoustic modality is more powerful, more efficient, because you don't have to have line of sight. You don't have to be in the visual field. It had been much less studied. As for faces and bodies, there is a voice area in the brain. There's a part of the brain, in this case in the temporal lobes, because it's the auditory system, in the superior temporal sulcus, which is this is the superior temporal sulcus, that responds more to voice than any other stimuli. Responds more to speech, but also to the so-called non-linguistic vocalizations like screaming, laughing, yawning, coughing. And it responds more, again, like the face and body areas, it responds more to human voice than to non-human voice. In humans, and there have been some studies recently in monkeys showing the equivalent. That is, they have a voice area, and this voice area responds more to monkey vocalization than to human vocalization. So there seems to be some species specificity. So these non-linguistic vocalizations seem to have the brain has the same machinery. The amygdala responds to them. The amygdala responds to non-linguistic vocalizations, to sounds of happiness. This is laughter. This is pleasure, sexual pleasure. We had some stimuli. Don't ask me how we got them. Crying for sadness, and fear for screams of fear. And you see that the amygdala responds to all of them. And if you pay attention, you see that at least in this case in the right amygdala, in fact, there's a stronger response to positive than to negative stimuli. And that's a bit counterintuitive because we've been talking about fear and the amygdala being involved in threats and all that. Yet, it seems that it does respond to threats, but it also responds to positive stimuli. And if anything, the magnitude is stronger. So that at first sight, one could say, well, maybe that's the difference between the auditory and the visual system, that in the visual system is about fear, and in the auditory is about other emotions. So we decided to go back to the literature and look at the visual literature. And it's true that if you go and you count the studies that report amygdala activation for fearful faces, is much larger than any other studies showing significant amygdala activation for happy, anger, sad, or disgust. So you say, yeah, okay, so the amygdala does seem to prefer fearful faces. But if you forget, instead of doing this, which is usually known as in sort of in the meta-analysis literature as a vote counting approach, that is, you just, for each study that says something, you tick the box and then you count. You say, well, of all those studies of these studies that did show an amygdala activation for something, let's look at the effect size. Let's look at the magnitude of what they reported. And if you do that, that's what you get. In fact, the magnitude is similar for all the negative emotions, and if anything, is larger for the positive ones. And you can, and this is not a bias because you can take the studies that did both positive and negative to make sure that there's no issue of statistical analysis, and you still get the effect. So what you have here is a bias in the literature, and is a bit of is sometimes known as a self-fulfilling prophecy, in that if you do a study with fearful faces and you don't have amygdala, you don't publish, period. So you'll find it. And there are many ways of finding, massaging your data in fMRI to find it. Whereas if you do something with sadness, you'll have the insula, which is an area important in empathy and sadness. You may have the amygdala, you may not. You'll publish it anyway. Because in the discussion you'll say, we didn't find amygdala activation contrary to such and such, but in line with such and such, right? And the reviewer will be happy. So there seems to be, and then there'll be another one for the fear. So the next one will, so we have a bias. But if you look at now the effect sizes, that's a different story. So this is very important when you read in general everything, but neuroimaging fMRI, there's a big discussion of the statistics involved, particularly I was telling you earlier about the so-called multiple comparison problem. We have so this is the approach has been changing, but the approach that we take here is so-called a massively univariate approach. You do a t-test for each voxel separately. So you do 100,000 or to 200,000 t-tests. So obviously if you do a 0.05, that's not a very efficient, because that means that 5%, roughly speaking, 5% of your voxels are false positives, and 5% of 200,000 is a lot. So you need to correct for multiple comparisons. If you use Bonferroni, which is the traditional one, you get nothing. It's very, very strict. So you have to, there are many ways of doing these things, and different people use different. So it's important to read the methods, even if you don't understand much of it, at least to see what sort of threshold they use, what type of statistic they use to avoid falling into this trap. Okay? So let me just spend 2 minutes. One last thing that I want to show you. And this, yet another way of expressing emotions, which is equally powerful, but has absolutely no evolutionary meaning, has no biological relevance whatsoever. Yet, it can produce emotions. So one of the questions that you can ask is, okay, we established that the amygdala is not just for fear. Some people have then proposed that it's so-called a biological relevance detector. So things that are survival related, biologically related, that's what the amygdala responds to. And we say, well, music is not. So that's what happens with music. So in fact, music is defined as a way of expressing emotions. There have been some studies showing that the amygdala is related to emotional recognition, but most of the studies use real pieces of music, and they divide unpleasant and unpleasant. So what we wanted to do is to see whether we can take the same approach as we took with faces, with bodies, and with voices. That is to have these basic emotions, have very short stimuli that are non-recognizable, they're unfamiliar, and yet they express the emotion. And this was, it's based on work done by Isabel Perez, one of the co-directors of Brahms. And they developed a database of musical excerpts that are based on the Western rules and are based, but they're created for research, so they're unfamiliar to subjects. And you can use very short pieces and express the emotion. So just to give you an example, I think it's, I don't know if you can hear it, but. So these are the equivalent of, you could say, well, you know, they're really stereotypical. Well, that's what facial expressions also are. These are very stereotypical way of expressing emotions. And they're very short. They're about a couple of seconds. And people are very good at recognizing them. So if you do a standard sort of valence and intensity arousal, you get exactly the same pattern for music that you get for vocalizations and for faces. So they're equally well recognized, they're equally well categorized, and so on and so forth. Let me quickly ask two questions. The first one is there a music area of the brain? It would make no sense because the brain hasn't evolved to process music. Well, there is. There's a part of the brain in the temporal lobes, in the superior anterior aspect of the temporal lobes, that responds more to music than to anything else. It still responds to other type of stimuli, but it responds more to music. And this is with piano and violin, but we just recently showed, this is work by my master, soon to be hopefully PhD student, showing that you can use a very large variety, I can show you later, of musical excerpts, also very short, single instruments. They have nothing in common in terms of frequency or anything, and voices from different languages. And it still responds more to music than to voice. So there seems to be the machinery there to do this. As I predicted, I would run out of so let me. So the question is, does the amygdala respond to emotions expressed by music? And the answer is yes. And it responds to, again, in a graded fashion, it seems to be a function of arousal. That is, it responds more to fearful than to happy compared to neutral and sad. It responds very little to sad. Neutral, as you may have heard, is what they call peaceful. So it's mildly, it's a mixture between sort of sad but also slightly relaxing type of music. So you could say it's mildly positive. And so it makes sense that it's here. So you have the amygdala that responds to particular these type of expressions. So the final question is, so I started saying that the amygdala is a very complex structure, and the problem with fMRI is that we cannot say anything about that complexity. So we're talking about the amygdala. But this is the same amygdala, is the music amygdala, the same as the voice amygdala, is the same as the face amygdala. So we cannot look inside at the subnuclear level, but at least we can see whether in the same subject, in the same paradigm, we get amygdala activation and how that amygdala is related. And the reason for that is that although the amygdala is a small structure there, if you take the peaks, that is the voxels that people report as being the amygdala, that's what you get. So the good news is that the center of gravity of this is in the amygdala. The bad news is it basically covers the entire temporal lobes. This is just, we think it's an error that the coordinates are in positive and negative, and I think that we think they put a positive and should put a negative, but it's just to illustrate. But here, you have a very broad sort of range of things that people call the amygdala. So it could be that this point here responds to voices, and this point here responds to music, and this point responds to face. So maybe it's the amygdala, but maybe it's not the same amygdala. So what we did, what one of the students, William Obey, did is using these stimuli that are very similar in length and in the basic emotions, in the same subjects, in the same session, in the same scanner, with the same analysis, see if there's anything. And the answer is yes. There's a part of the amygdala, a little cluster in the amygdala, responds to fear, be it presented in the face form, in the music form, or in the vocal form. That is, it responds to fearful expressions regardless of how they are expressed. But this only works for fear. So, and there are other areas. So to sort of summarize, it seems that the brain has whether evolved or is developed or is prepared to develop during the course of lifetime, different areas that seem to be they're not, when people talk about a voice-specific or face-specific area, that's not true. That's also an important thing. It's voice-preferred because, as I quickly showed, it responds to other stimuli. It just responds more and more efficiently, potentially, to this type of stimuli. So and for the different ways of expressing emotions, for faces, for bodies, for voices, and even for music, there seems to be. And the amygdala responds to all of them, and there are similarities and differences. So they seem to respond, the amygdala seems to respond to emotions in general, sometimes more to one of theirs. But once I started talking about fear, the amygdala and fear, and then I said, well, the amygdala is not about fear. And I'm finishing in a way that, yes, the amygdala is fear. Because although the amygdala is not just fear, fear seems to be important. Fear seems to be unique. I didn't show you, but there's an enhancement of memory for emotional expressions that it differs across different modalities, except for fear. There's always an enhancement of memory for fearful expressions, regardless of how they're presented. And there's an amygdala response to fearful expressions, regardless of how they're presented, and it seems to be in the same cluster. We don't know if it's the same neurons, and that's what we're doing now using so-called adaptation paradigm. So it seems to be something special about the amygdala and fear. Right? If you want to know more, you can read a little paper. We have a book on everything, but including sort of some of the stuff I work. And we've been interested lately in the relation between music and speech, and this is where with Isabel Perez, and we're doing some studies to try to disentangle a bit more about the representation of music and the relation between music and speech. And of course, this is work that's really done by other people and paid by taxes from my fellow Canadians. So that's it. Sorry, I went a bit over time, but I was I told you I would. So the question, I'm supposed to repeat the question. So the question is whether so that I think I'll rephrase it. That's the advantage. In two questions. One is there are different sizes of amygdala, and the other one is, as has been suggested with the hippocampus, that there can be neurogenesis, there can be growth of the hippocampus, of the amygdala. So in terms of sizes, yes, less. So there's a lot of work on the hippocampus. And for example, the PTSD literature there's a huge literature on size of the hippocampus on PTSD, and the idea is that individuals with PTSD have smaller hippocampi than controls. And at the time, it was interpreted based on Sapolsky's work on stress and the hippocampus, that elevated stress damages the hippocampus and kills neurons. So the idea was trauma basically kills the neurons of the hippocampus. But Roger Pittman from Harvard, he wasn't very convinced about that, and he always suspected that it was more of a risk factor than a consequence. And he proved this to some extent in a very incredibly elegant experiment using twins, identical twins. At the time, only one identical twin will go to Vietnam. One was spared. And so one was exposed to trauma, had PTSD, the other one stayed at home. The one with trauma with PTSD had smaller hippocampus, the twin brother as well. And they were actually correlated. And you can show that there's neurogenesis. So the hippocampus seems to be more flexible. So the amygdala, there are studies suggesting there are differences in volume in the amygdala, but it's much less clear. The only one that it seems to be really consistent is that individuals with autism have bigger amygdala. And in terms of neurogenesis of growth, I'm not aware of any study having shown that. So we don't know whether that's the case or not. So kind of the short end is that yes, structural differences there are, but they're much more, perhaps because the amygdala is a smaller structure, but they're more subtle, if any, and they're more varied than with the hippocampus that's more clear cut. There are many more during there are very lots of studies showing that during the course of life the hippocampus shrinks, and it shrinks more in men than in women and more rapidly. So and that's been associated with the loss of memory and so on and so forth. In the amygdala is less clear, that story. So the question is, what about singing, which is both voice and music? And exactly we thought exactly the same way. This is so the part of the work of music is done in collaboration with who mysteriously disappeared. Oh yeah, Luis Concha from Mexico, and he had a singer. He recorded a singer singing basically sentences. And he was going to run the experiment, and I'm not sure he did, but if he did, I don't know the results. So I'm waiting. But yes, that's exactly the question we had is it's voice. But the important, so one thing that one of the slides that I skipped is that the music area also responds to voice. It just responds more to music. So the assumption would be that it would kind of be even stronger response potential than even musical instruments. Again, one of the things that I didn't show, and we're still working in some publish, is that it seems that part of the brain, what seems to be responding is to rhythm, what's sometimes known as pulse clarity. So that is how rhythmic the stimulus is. And it responds to how rhythmic the stimulus is regardless of whether it's music or voice. It's just that music tends to be more rhythmic than anything else, and therefore responds more to that. So in this case, again, singing voice would probably sort of be either an additive effect and you would get twice as much, or some sort of but yes, that's exactly the study we're looking at. The other question that people ask, and this is, I think Isabel is doing something, is singing in the scanner, sort of having, that's, as you can imagine, very complicated because singing involves moving, and this is the equivalent of taking a 20-minute picture of somebody. So you don't want them, really don't want to move because we're looking at, talking about millimeters. So they've been training, some people have been training singers to sing without moving, sort of really just moving their mouth without sort of really doing it, and they can do it. And then after that training, in fact, he's not there, but Robert Zatori, who's the other co-director of Brahms, he's now also having, his students are teaching people to play instruments. So they created a piano and a cello that is magnetic compatible. So they go into the scanner and they do that. So maybe I shouldn't say that because I'm on camera, so I don't want to spoil it, but yes. So the question is about the generalization of the responses of the amygdala to other potentially fearful stimuli, and how that can be, if any, if it can be controlled. And so yes, as you suggest, so the main inhibitory input to the amygdala in terms of controlling is the prefrontal cortex, particularly the medial prefrontal cortex, and that's why it's thought that in disorders like PTSD, they have, there's been, I told you about the exaggerated amygdala response, but also there's a hypofunctionality of the prefrontal cortex, and sometimes people have found problems with the connectivity. Stimulus generalization hasn't been studied much in humans, so we don't really know exactly how it operates and how it will be controlled. But the assumption is that it would be the prefrontal and potentially the hippocampus as well. At some point we hypothesized, whether we never proved it, but that the hippocampal input to the amygdala will provide contextual information. So the idea is, you see a snake and the amygdala responds, but it's very different to see a snake when you're walking in the woods than to see a snake in the zoo behind a glass. It's exactly the same snake. Yet, in one case, it makes sense to respond, and in the other one it doesn't. And one of the problems with phobias is that they respond regardless, and they respond even if it's a picture. And there's very little known. There have been some studies with phobia, but it's not exactly know what is causing this generalization, but the assumption is that either it's the hippocampus providing the contextual information that in one case is safe and the other is not, or the prefrontal cortex also inhibiting the inappropriate responses, and potentially in people with phobia this process would not work, and therefore they would have the response kind of in abstract, in absence of any sort of other information. They cannot take advantage of any other information that would help them integrate the stimulus with the context. Okay, so the question, maybe I shouldn't just say and ignore it, is about fear of falling. And the problem, in a more general sense, I guess, is the problem of the scanner. So as you say, the scanner is this tube where one has to be really still. So it limits a lot what one can do. And as I was saying, as example, these instruments. One of the ways people have tried to get around this is virtual reality. It's not this clearly, you don't have the proprioceptive information because you're not moving, but at least you could potentially be a little sort of avatar that is about to fall or something. I mean, that's why in a more general sense, fMRI is a great technique and it's really useful, but it has many limitations. And one of them, there are limitations in the temporal resolution that I can tell about, the spatial resolution, but from a practical perspective, there are two major limitations. One is lack of movement, and the other one is that it's a magnet, so anything that is not magnetic compatible cannot go in, so a lot of patients cannot go in, and the paraphernalia that you can have is very limited. So perhaps in situations like that, you can use other techniques. So there are techniques like EEG, electroencephalography, or NIRS, near-infrared spectroscopy, that also looks at the oxygen and deoxygenated blood, but instead of looking at the magnetic properties, looks at the optical properties. And these are much more portable. In fact, people have developed wireless systems. So you basically put a helmet, and you can get somebody sort of free in the world, and you can measure their responses while they're falling off a cliff, or whatever is that the experiment that you do. And so potentially for those type of studies, the problem with these techniques is that they record mostly cortical activity. So if you're interested in the motor system, for example, that's perfect. If you're interested in the amygdala or subcortical structures, then it's a bit more difficult because they basically the NIRS cannot look beyond the cortex because the optical system doesn't work. And EEG, although you can reconstruct the sources, is more difficult. So there are some limitations on these techniques as well. But yeah, it's a trade-off on what you want to do and you can do. And this is going back, one of the criticisms that we always get about this, that this is not real emotions. And well, is what you can do in a scanner. So you have to have you have to translate your goal into something that you can measure. And if your experiment can be great, but unless you say, well, once they invent the portable scanner that I can sort of wear in my head and walk around, then I cannot do my experiment. Well, then you can't. So the question is, how many voxels is the amygdala? And I deleted that because, but it's I had that slide. You could do the calculation, but it's very few. If you do depends on the voxel size, because you can use different voxel size. But if you were to do a standard experiment in which the voxel is 3 by 3 by 3 millimeters, the central nucleus of the amygdala would be less than 1 voxel. Just to give you an example, the amygdala would be 20 voxels or so. That's why, but because of we use spatial smoothing and things like that, in fact, you get a larger amygdala. But and then there's the concept of the extended amygdala, but it's a very, very small structure, and that's why this is no way we can resolve, at least with the techniques we have now, this so-called this nuclei. People have argued that they can detect you can divide the amygdala in two large, the sort of the basolateral sort of complex and the corticomedial complex. And people have argued that they can tell them apart with fMRI. I don't think so. It's debatable, but it's anywhere between 20 and 100 voxels, depending on how you measure it, how you do it. But it's very, very small compared to the thousands of voxels that you will get when you do visual cortex or motor cortex or things like that. Yeah. Yeah. So the question is kind of a counterintuitive idea that I was saying. Maybe I shouldn't say it because I haven't read the literature, but that autism is associated with larger amygdala, yet individuals with autism have problems recognizing emotions. So it's one of the things. So autism in general is associated with larger brains, with larger connectivity. So and one of the theories is that when we're born, we have many more connections than later on, and there's pruning of information. And in individuals with autism, there's less pruning. They have more connections between everywhere in the brain. And so it's possible that's why their amygdala are bigger, is simply because it hasn't kind of fine-tuned, it hasn't pruned. And this is another, it brings another issue, another question. There's a lot of studies. In fact, we did some looking now at structure and saying there are differences in the volume between, I was talking earlier about PTSD. And it's always the assumption that bigger is better, And the answer is, in some cases, yes, but when it comes to the brain, we don't really know. Is it really larger, meaning better functioning, or is there too much noise? It could be. So in fact, there are a lot of studies showing that training reduces the signal rather than enhances the signal. And the explanation is in terms of this. I was showing you a little bit of this tuning receptive fields, and the idea is that it becomes more efficient. So when something that takes more work and it requires more neurons, and once you train, then you find an optimal way of solving that problem. And by doing that, you use fewer resources. So the relation between size and signal is not clear. And even the relation between signal and real processing and efficiency is not clear either. Again, I'm assuming, by telling you all this story, I'm assuming that bigger signal means better, but we don't really know, because means that there's more blood flow. There's more blood oxygenation, which probably means that there's more neural activity. Is more neural activity better? No. Tell that to somebody with epilepsy. Clearly not. So, it's now I'm sort of shooting myself in the foot of my entire talk, but you can cut that later. But it's a complex. So in a less depressing way, it's also sort of a word of caution that one has to interpret these things. One has to know what the limitations of these different techniques are and how to interpret the results. On their own, they don't mean anything. You need to try to put them in context and have other sources of information to do that. But the short answer is, yeah, I don't know.